Bismillahirrahmanirrahim. Now we will start the chapter nucleotide metabolism. As we know that ATP, ADP, GMP, CTP, CDP, UDP all are the nucleotides. These nucleotides are very important for life and the biochemical processes taking place within the cells. These nucleotides are the basic components of the genetic material DNA. They are also the components of RNA and protein synthesizing machinery. They also have a role in cell proliferation. The nucleotides are the structural components of coenzymes like coenzyme A, NAD, FAD, NADP, S-adenosyle methionine, etc. The nucleotides like cyclic AMP, cyclic GMP, they act as second messengers and play a key role in signal transduction pathways. ATP and ADP are also the principal players in the energy transduction processes. They are also the principal donors and acceptors of the phosphoryl groups in many metabolic pathways. They also participate in metabolic reactions like protein synthesis, lipid synthesis. The nucleotides regulate the enzyme activities, especially the enzymes whose activities are activated by phosphorylation and dephosphorylation. These nucleotides also take part in many metabolic processes concerning sugar, carbohydrates or lipid molecules. So in these metabolic pathways, the nucleotides activate the sugar molecules or the lipid molecules like UDP glucose, UDP galactose or CDP acyl glycerol. The synthetic purine and pyrimidine analogs, they contain halogens, thiols, additional nitrogen atoms. These synthetic analogs are used in cancer chemotherapy and they are also used as immunosuppressants in organ transplant rejection reactions. So these are some biochemical or the biomedical importances of the nucleotides. As we know that nucleotides are composed of three components. One is the nitrogenous base, other is the pentose sugar, that is ribose, and then the phosphate group. The combination of nucleic acid with a pentose sugar is called a nucleoside. The nitrogenous bases are of two types. One is the purines and the other one is the pyrimidines. Purines are adenine or guanine. These purines are present in both DNA and RNA. The pyrimidines, that is cytosol, thymine and uracil, out of which cytosine is the pyrimidine which is present in both DNA and RNA, while thymine is present in DNA and uracil is the part of RNA molecules only. This diagram shows the basic ring structure of purines and pyrimidines. As it is evident from the diagram that purine are the larger molecules having two rings structures. One is the imidazole ring and the other one is the cyclohexane ring, while the pyrimidines have only the cyclohexane ring. The purines adenine and guanine In these purine nucleotides, the purine ring is common to both, and they differ in having the different functional groups attached to them. Similarly, the pyrimidine nucleotides, cytosine, thymine, and uracil, their basic ring structure is common, and they differ only in having the additional function groups which are attached to these to this pyrimidine ring structure. Now the biosynthesis of purine nucleotides. The biosynthesis of purine nucleotides, there are three processes that contribute in their synthesis. One is the synthesis of nucleotides from amphibolic intermediates. The other process is the phosphoribosylation of purines. The third one is the phosphorylation of purine nucleosides. The purines are synthesized in most of the tissues, but the major site of their synthesis is the liver. De novo synthesis is the major pathway of the synthesis of purine nucleotides. In the synthesis of purine nucleotides, in this pathway, the synthesis of purine nucleotides is from various small molecules which are derived as intermediates of many metabolic pathways. Salvage pathway is the minor pathway, and this salvage pathway is basically the recycling of the degraded purine bases or the nucleosides. Now this is the ring structure of a purine, and this diagram shows the sources of the atoms. In this diagram, it can be seen that the atom 4, 5 and 7, these are derived from glycine, and the atom number 6 is provided by the carbon dioxide. The atom number 1, that is nitrogen, is derived from the aspartate. The atom number 2 and 8, these are derived from the derivatives of tetrahydrofolate. One is formyl tetrahydrofolate and the other one is the methinyl tetrahydrofolate. The nitrogens which are present at the 3rd and 9th position, they are derived from the glutamine. So it is clear from this diagram that in the synthesis of a purine ring, 3 amino acids are involved. One is the glutamine, other one is the glycine and the aspartate. And the 2 atoms are provided by the tetrahydrofolate derivatives, and the one comes from the carbon dioxide. So this is the sources of atoms for the synthesis of a purine ring. Now the parent purine nucleotide which is first synthesized is the inosine monophosphate. This inosine monophosphate is composed of a purine base hypoxanthine, a ribose and a phosphate group. From this inosine monophosphate, other nucleotides are synthesized like adenosine monophosphate or guanosine monophosphate. For the synthesis of purine ribonucleotides, ribose 5-phosphate is the starting material. This ribose 5-phosphate is produced in hexose monophosphate pathway, that is the HMP pathway of carbohydrate metabolism. The first of all, this ribose 5-phosphate, it reacts with ATP. the ATP transfers its pyrophosphate group to the ribose 5-phosphate. So as a result, the phosphoribosyl pyrophosphate PRPP is produced. This reaction, the synthesis of PRPP, is catalyzed by the enzyme PRPP synthase. This enzyme is inhibited by the PRPP as a feedback inhibition. Now, this PRPP phosphoribosyl pyrophosphate, it reacts with glutamine. The glutamine, the amide nitrogen of glutamine replaces the pyrophosphate from this PRPP, and as a result the PRPP is converted to phosphoribosylamine, as clear in the diagram that this pyrophosphate group is replaced with amino group that comes from the glutamine, and this reaction is catalyzed by the enzyme PRPP glutamyl amido transferase. To remember, amido transferase. This enzyme PRPP glutamyl amido transferase is also controlled by feedback inhibition by the nucleotides IMP, AMP and GMP. In the next step, this phosphoribosylamine, it reacts with glycine. This is basically the condensation reaction in which the glycine condenses with the phosphoribosylamine. So, as a result of this reaction, glycinamide ribosyl 5-phosphate is produced. This reaction is also energy requiring process in which ATP provides the energy. So, in this reaction, glycinamide ribosyl 5-phosphate is produced. Then in the next step, the methynyl tetrahydrofolate, it provides the formyl group to the glycinamide ribosyl 5-phosphate. Then the enzyme catalyzing this reaction is formyl transferase. So as a result of this reaction, the formyl glycinamide ribosyl 5-phosphate is produced. These initial reactions which are explained are important one because the enzymes taking part, the enzymes catalyzing these reactions are subjected to the regulation of the purine nucleotide biosynthesis. Then in the next step, the nitrogen at number 3 is provided by the glutamine. Then after the attachment of this nitrogen at the third position of the purine ring, the initial ring is closed, that is the imidazole ring. That is clear in this diagram, that this on this ribose 5-phosphate, the atom number 9, 4, 5, 7 and 8, these form the imidazole ring, and this closure of the ring takes place after the attachment of nitrogen that comes from the glutamine, and this nitrogen that is provided by the glutamine is the third carbon of the purine ring. Then after the closure of the imidazole ring. In the next step, the carbon atom which is present at the sixth position of the ring is provided by the carbon dioxide. Then the nitrogen that is present at the position 1 of the ring is provided by the aspartate. And the aspartate, after providing the nitrogen, amino nitrogen to this ring, the carbon structure is released as the fumarate. Then after the bonding of nitrogen from the aspartate, then in the next step, the formyl tetrahydrofolate, it provides the formyl group, and then after the formyl group attachment, the ring is closed, that is the cyclohexane ring. So after the closure of this second ring, the compound which is formed is the inosine monophosphate. So in this way, the parent nucleotide inosine monophosphate is synthesized. Now this is the summary of the steps that summary of the steps of the purine synthesis. In this chart we see that at the first step, glutamine provides the nitrogen at number 9. Then glycine provides the atoms at 4, 5 and 7. Then the methynyl tetrahydrofolate provides the carbon at 8. And then glutamine provides the nitrogen at the position 3. And then the ring is closed. After that, the carbon dioxide provides the carbon at number 6, and then aspartate provides the nitrogen at number 1, and then after the removal of the fumarate, the formyl tetrahydrofolate provides the carbon at number 2, and then the ring is closed. So this is the summary of the reactions for the synthesis of inosine monophosphate. So thank you.